All right, my friends, so this is the rat dissection. If you are sitting here going, oh yeah, I got this, particularly you seniors, if you're going, oh yeah, I know what she's talking about, you're going to be sitting pretty for the exam. If you are like, oh, I had no clue what she's talking about, oh yeah, that's distant memory, you need to do some serious studying. Okay, so here we go. So this is a rat. This is a mammal. And this is a mammal I know for two reasons why it's a mammal. The two distinct reasons are it's got hair and it's got mammary glands. They're the two characteristics that set this animal apart from the rest and make it a mammal. So this has hair. In fact, it has modified hair all over its body. It's got fur on the outside. This fur on the outside is going to provide it with insulation, protection. There are many things that the hair does for a mammal, right? Camouflage, sensory. Well, we can see the sensory. You can see these whiskers, these vibrissae. These are the whiskers that are modified hair that is going to allow for the rat to pick up vibrations and can size and can do all sorts of things with these whiskers. Very good. The hair over the body, very important. I'm going to flip mine to the ventral side up, and you'll see that there are nipples. These teats, these are the mammary glands. They're going to produce milk for the purposes of lactation, for the purpose of nourishing the offspring for the developing babies after they are born. Okay, so let's go back to the position that it was in, which is the dorsal side up. I need to make sure you know your positions. This is the dorsal side, that's the back. What it's lying on, and where I saw the mammary glands, that's the ventral side. The tail end, that's the posterior end, coming in from this side, that's posterior, and coming in from the head is the anterior. What we've also looked at is that we've looked at a number of cavities. Okay, so I'm going to flip this rat over a second so we can have a look on the outside. On the ventral side up, it's easier to see the cavities, remembering that a cavity is a hole in which organs reside. Okay, so there is the cranial cavity, which contains the brain. It's lying on the spinal cavity, which obviously contains the spinal cord. But on the ventral side, we can see it's divided into three main cavities. This region right here, where I've got my ribs and below the cranial cavity, this is the thoracic cavity. It has a definitive end stopping point. That's the diaphragm. A diaphragm is a flap of muscle that is used for separating the thoracic from the abdominal pelvic cavity, and also obviously used for breathing. Now, below my finger, this area here, this is 2-in-1. This is the abdominopelvic. And if I put my fingers here, about the level of the hip, anything in between my fingers in this region is abdominal. Anything below is going to be pelvic. So pelvic, abdominal, thoracic, cranial. Okay? Okay. Now, let's go to the posterior end, and I can see the tail. Now, this is the fifth appendicular portion. Wow, I had a mental freeze. Here I go, the fifth appendicular portion. These are the appendages, anything that gets added on to the outside of the body. Okay, the axial portion is if I chopped off all of the arms and legs and the tail, so that would be just the main trunk of the body. That will be the axial portion. So we're going to go to the most posterior part, which is the tail. And I want you to run your fingers down the length of the tail from the anus to the tip, and then gently run it the opposite direction from the tip towards the anus. Gently, go ahead. And what you're going to feel on there is that you'll feel a different sensation of hairs that are on this tail. Again, hair is a purpose of being a mammal. Now, this hair is made up of a specific protein called keratin. And that is, the hair is in one direction. If you notice, if you go from the tip towards the anus, it feels rough. And if you go from the anus to the tip, it feels smooth. That is because the hair has directional flow. and it allows for the air to transfer over the body of the rat and make sure that, you know, it gives it signaling of where things are. Another thing that the tail does is it counterbalances. So as the rat is running, the tail is used to counterbalance its forward locomotion. And it helps it, if it wants to lean up and grab something, that can be used as a rest. It's not like a prehensile tail, which is used as an extra arm in certain primates, but this tail certainly does have advantages. Why don't we have a tail? We don't need one. And because we are bipedal, meaning we're walking on two legs upright, that tail would just get in the way. This is a quadruped, meaning that this rat is going to walk on all four. Okay, any questions so far? I'm going quickly. I'm going to look at the posterior end, and I'm going to have a look at the bottom region, okay, for the genital region. So the first hole that you can see that is right here, this is the anus. This is where the rat will defecate or poop out of. Go ahead, you can have a look at yours. Then if you go a little anterior, going forward, closer to the ventral surface, you'll see a larger hole. That is the vagina. And then in front of that, you can see there's a little projection. This is the protective area, a papillae, that is going to protect the urethra where you urinate out of, okay? Anus, vagina, urethra, okay? Happy? Any questions? I thought I heard, oh, my clock has been returned. That's very kind of them. All right, so here we go. So obviously we need to have a hole for babies. That's the vagina. This is pee, this is poop. This is the exit area for the animal. It is in the opposite end to the entrance, right? So we've got a division, a large division between the excretion and the input, because you don't want to mix the two. You don't want to put waste back into the body, okay? When we look at this rat, we can see that we are going to explore the 11 systems of the human body. They are run, Mr. Sliced. Respiratory, urinary, nervous, muscular, reproductive, skeletal, lymphatic, integumentary, cardiovascular, endocrine, and digestive. And because this is the first whole organism, we'll be able to see the hierarchy of life in play. Obviously, we're not going to see it from the lowest level of atoms that come together to form molecules, that come together to form macromolecules, of which there are 4: proteins, lipids, carbohydrates, and nucleic acids. But those are going to come together to form the macromolecules from organelles. Those organelles form cells. That's the basic unit of life. Those cells come together to form tissue. And we'll see various different types of tissue. There are 4 different types of tissue. We have epithelial, connective, nervous, and muscular. I'm going to show you muscular in a minute. And then we have the tissues come together to form organs. Yes, that's what we're going to see. How these organs then form the organ systems that make up the whole organism. Poof! All right. I hope you're seeing I'm doing a lot of review here as I'm going along. I know we haven't even got into the rat yet, but this is really important stuff. Okay, so we've done the posterior end. Let's go to the cranial cavity, to the anterior end. And what we're going to look at is we're going to have a look at these lovely little ears. Now, these ears, the outside of the ears, is a really cool word, the pinnae, P-I-N-N-A-E, pinnae. These are the ears. Now, these ears are slightly different than our ears. Why? Because they can move. Now, some of you might be able to wiggle your ears. I cannot. But you don't move your ears for directional input of sound to find out where is that noise coming from. We have different sensory inputs. But these ears, the pinnae, P-I-N-N-A-E, these are going to move in order to funnel in the sound into the rat's auditory canal so it can get processed in the brain, okay? One of the senses, remember there are 5 senses: touch, smell, hearing, sight, and taste, okay? This rat actually has an extra one, which is the whiskers, part of the touch, if you will. These whiskers, the vibrissae, these are going to be here to pick up electrochemical vibrations and also for sizing. You've never seen a rat get stuck in a hole, have you? No. Why? Because it pokes a little head in, and those vibrissae, the whiskers, bend back, and depending upon how far they bend back, it's going to determine if the rest of their body is going to go through, because they're a vertebrate. They've got lots of bones in it. They're not going to be able to squeeze through a little teeny tiny hole like an octopus can, right? Okay. The next one we've got is our sight, and these are our eyes. And you can see that there is an enormous amount of protection on our eyes. We went in great detail about our eyes. We looked at how light is just received by the eyes. It goes in through the eye. We've got the cornea on the front, the iris, which is going to regulate the size of the pupil, which is quite literally a hole. The light then goes in, is focused by the lens. It goes into the back of the eye, onto the retina, onto 2 specialized photoreceptor cells called rods and cones in black and white and color. And then they're going to get transferred down the optic disc to the brain, where it crosses over at the optic chiasm, and it goes to the occipital lobes of the brain in order to get processed. Now, the eye itself has got lots of protection, and we talked about this before. We talked about eyelids, eyelashes, eyebrows, the tears. We also talked about how the eye, the white part of the eye, is a really tough structure called the sclera. And this is going to help to hold the eye and keep and maintain its shape, okay? So again, the eye is just the receiver of light. It doesn't do anything but receive light and sends it as an electrical signal to the brain to get processed, and that's what you're using right now to see. The next thing that we can see are our beautiful little nose. Oh, it's so cute. Are you going to focus, dude? Focus, dude. Why are you not? Oh, don't be like this. There we go. It's a bit better, isn't it? So here we can have a look at the nose. You can see that the nose is divided in half by a septum. This nasal septum separates the 2 nostrils, which are called the nares, N-A-R-E-S. Nares, N-A-R-E-S. These are the nostrils, and they're separated down the middle by a septum. Remember that a septum divides a cavity in half. Okay. The nostrils here are going to take up the smell, and it is going to go and get processed by the olfactory bulbs inside the brain. So what I'm hoping you're understanding is that the information gets inputted into the brain, and the brain takes that information and then processes it and responds to what it needs to. So it receives information, interprets the information, and responds to it. That is the purpose of the nervous system, okay? So nose smelling. Now, sense of smell is very, very important for the next one, which is the sense of taste. Now, we did a whole experiment at home about our sense of taste, and we saw that there is experience and exposure, genetics, if you will, that are going to influence your taste palette, how you taste things. There are 5 main tastes: salty, sweet, bitter, sour, and umami, which is the savory taste. The taste is done on the tongue, and then that information is again sent to the brain to be processed. You don't actually taste with your tongue. You receive the information on your taste buds, which gets transferred to your brain. Your brain then interprets it and sends it back as a, hey, this tastes good, this tastes bad, okay? Now, I am going to talk one more thing before we get into the oral cavity, and I'll tell you more about that in a second. I wanted to have a look at these limbs a second, these axial portions, the appendicular portions coming off the axial. These limbs, there are 4 limbs, and the forelimbs and the hind limbs are different. You can see that these hind limbs are incredibly muscular and strong, whereas these forelimbs, these ones most anteriorly, they are less developed. Why? Because again, look at this. Anatomy dictates physiology. The structure determines the function. The structure of these hind limbs are much more powerful because they are going to be the locomotion. They're the ones that are going to move the rat. These forelimbs are smaller. They don't need to be robust and muscular. They're going to be there for steering. So as these back wheels, if you will, are moving, the front ones are telling which direction to go in. They're also responsible for bringing the food to the mouth, to the oral cavity, or eating, okay? Any questions so far? Okay, so what I'm going to do now is I'm going to show you how to do the Joker's cut, okay? Now, this is an important incision that you need to know how to do, okay? I'm going to just leave it at that for a second. How you do a Joker's cut is you are going to see that this is the lower jaw. That is the lower jaw, remember, is the mandible. The top jaw is the maxillae. You are going to want to move the mandible, the lower jaw, away from the maxillae to be able to look inside the mouth. And to do that, I'm going to look for the corner of the mouth, and I'm going to look at the angle to the bottom of the pinnae of the ears. I'm going to make an imaginary line in my head, so it's going to go like this on this side, and I'm going to cut it on the side, and I'm going to cut it through the jaw. Now, watch what I'm going to do, and then I'm going to tell you about yours in a second. So what I'm going to do is I am not going to go past the eye, because if I go any further, I'm going to go in the cranial cavity into the skull. I do not want to do that. I just want to get into the oral cavity. So I'm going to leave it on its side, and I'm going to place my scalpel at the corner of the mouth and about just in line with the ear and just below the eye, and I'm going to cut through the jaw. I'm going to flip it over and do the same thing on the other side. Now, what you may have heard was a little crunch, and that is because the bones of this particular organism are calcified, hardened. If I were to do this to the babies that are inside my rat, it would cut through like a knife through hot knife through butter, because a baby's bones are not hardened until well after they are born, because it allows for them to get squeezed through the birth canal. So again, from the ear, a line to the corner of the mouth, below the eye, I'm going to insert my scalpel and crunchy, crunchy, I'm going to cut through. Well, that's because, again, my rat's bones are hard. I'm going to grab hold of the lower jaw, the mandible. I'm going to pull it away from the maxillae, and I'm going to be careful of the dentition, the types of teeth that are in here. This is where I find out I haven't cut it deeper. All right, so I'm just going to do it a little bit more. Now, if my scalpel were to break, I would say code 1, and I would just take my hands out the tray, and I would be safe in that mechanism. All right, so let's give this another try. Lower jaw, which is the mandible, pull away from the maxillae, which is the top jaw. And I can open up. It takes a little bit of force, but I've now opened up the jaw. Now I can have a look in the oral cavity. This is the Joker's cut. Now, I was kind. This morning I got here early, and I have pre-cut your Joker's incision. So there should only be a need to grab the lower jaw and pull it away from the top jaw to open it up. So go ahead right now. You've got paper towels afterwards to wipe your hands. Go ahead, grab, I've already cut them for you. I hope I've cut them well enough. Grab the lower jaw, time out, quiet, Hey, we're not talking. This doesn't work if you talk. Grab the lower jaw, the mandible, and pull it away from the maxillae. If you cannot do it, raise your hand, I'll be there. Okay, but give it a try. There's a difference between can't do it and don't want to do it. All right. If it doesn't come easily, give it a good pull. And if it doesn't come easily, then I may not have cut it through far enough. Okay?
I don't think you have to.
I
Sorry.
I will never do it. If it hasn't been all the way through, you can always recut it if you want to.
Should we flip our scribbles or something?
No, I would leave it ventral side or the oral cavity.
Oh.
Make sure the head, if you're cutting it, is flat on the tray sideways. This is where gauge and thumb are going to Okay. Okay. So we don't Oh, God. Okay, great. Yeah, I agree. All right, so have a look on the inside. Coming. All right, have a look on the inside. You'll be able to see that on the inside of the mouth, you should be able to see quite clearly that there is a variety of different teeth that are there. You can run your finger along the jaw lines and you'll be able to feel the different dentition. Remember, dentition is the collection of teeth. You'll also see the tongue. The tongue is there and it's really a very clever little muscle. Why is it so clever? Because it is only attached at one end. Most other muscles are attached at both ends. And this tongue is only at one end. It's also very interesting. We're talking about muscles. There are 3 different types of muscle. The 3 types of muscle are cardiac, which you can only find in the heart. You've got skeletal muscle, which is attached to the bones. And then you have smooth muscle, which is in digestive organs and in blood vessels and the like. Cardiac, skeletal, and smooth. Blood vessels, like a bladder. Anyone else? We're all good. The other thing that I want you to have a look at is as we go into this, you're going to look at the roof of the mouth. And you are going to see that there is a difference between the roof of the mouth and further back. If we look at the roof of the mouth, can you all see the roof of the mouth? Oof. I'm all hot now from doing that. All right, so here we go. If you look at the roof of the mouth, you can see that it is different from the rest of the oral cavity. I'm trying to get here so you can see. There we go. All right, so if you look at the roof of the mouth, you can see how, just like if you were to put your tongue behind your front two teeth and draw it backwards, you'll feel that there's hard ridges right here, and then it goes soft. These are the palates. This is the hard palate and the soft palate. The hard palate and the soft palate are used to separate the nasal from the oral cavity. It's also used to help with the formation of a food bolus, and it's also used in talking, in speech. Now, the part that's going to be very important for us to see is that there is as I go back past the soft palate, as where my probe is just here, that is the pharynx, P-H-A-R-Y-N-X. Fantastic Scrabble word, the pharynx. That is the space where in the back of the throat and the back of the mouth, it decides whether or not it is going to go down the food tube, the esophagus, or it's going to go down the air tube, the trachea, okay? So you have to make sure that the food goes down the food tube and not down the air tube. So on the trachea, remember, there is a flap of cartilage that goes over the top of the trachea when we swallow, and that is going to be the epiglottis that closes over the top of the trachea. And then behind the trachea, which is reinforced with rings of cartilage so it doesn't collapse, behind that is the esophagus, which is the flexible tube that allows food and water to go down to the stomach. The trachea is going to allow air to go down to the lungs. So that's the oral cavity. So we've got the maxillae, the mandible, the dentition, the tongue. We've got the palate and the pharynx. And last thing. So talking about chewing and eating and ingesting of food, we need to eat that food in order to supply our body with all of the nutrients we need, particularly that glucose molecule, and we need to breathe in that oxygen all in the same area, which is why you got two tubes in the same place, all of the input, right? Why do we need that? That is for cellular respiration. C6H12O6 plus O2 yields CO2 plus H2O plus ATP, which stands for adenosine triphosphate. Okay, remember that's the only word I've asked you to spell correctly. Now, when the food gets put into the mouth, into the oral cavity, there is both chemical and mechanical digestion that's going to go on, which is the mechanical, the teeth and the jaws are grinding it all up, moving that food around the palate. And then the chemical is the saliva that is going to get dumped into the oral cavity and mixing it all up so that you have mastication. Be careful how you say that. Mastication, which is the addition of saliva to the mechanical, so chemical and mechanical. In the saliva, don't forget that the chemical process is mainly done with the breaking down of carbohydrates, one of those macromolecules for short-term energy. Those, so the carbohydrates are going to be broken down by an enzyme called amylase. And what made your food bolus nice and shiny and smooth so it can be swallowed is that protein lubricant called mucin. Some of your food boluses were fabulous, by the way. I enjoyed looking at those. Yeah, I know I'm weird, but that's all right. Some of them, yeah, were dainty. Some of them were enormous. All right, here we go. So this is where we are going to cut open our organism. Okay, remember, make it so that it is comfortable to you. Remembering your scalpel rules, don't be leaning. Everything needs to be in front of you for cutting. I am going to demonstrate how to cut any quadruped. Okay, I'm going to demonstrate how to cut any quadruped. When I am going to cut down, I don't touch the scalpel till I tell you. When I cut down the midline, and I get through the thoracic cavity, as I'm getting through the skin and the muscle, I'm going to hit bone. I'm not going to saw through the bone just yet. That is going to be my guide. I'm then going to make sure I lift up on the skin so I don't cut the viscera, meaning the organs in the abdominal, pelvic, or thoracic cavity. So I'm going to make sure that I'm going to be cutting by lifting and cutting as I go. All right, so here I go. I'm going to take my scalpel. I'm running it down the midline. Yes, I hit bone. I'm not getting all the way through. That's okay. And then when I get past where the diaphragm would be, I want to make sure that I lift up so that I don't cut the organs underneath. This scalpel is not my friend today. And I am going to, there we go. I'm going to cut down through the organs, through the abdominal pelvic region. I'm going to come down to the urogenital opening. All right. Making sure I've gone up here. Now, this is where some of you may be a little bit like, what? I'm going to rotate it and I'm going to let it drain because it does have a little bit of fluid in there. Okay. That's okay. Just drain it. This is the only one that's full of fluid. Okay, so now what I'm going to do, oh, what's going on in here? Oh, poor rat. All right, fine. I'm going to now cut across at the thighs. I'm going to go horizontally now. Again, I'm cutting and looking where I'm cutting so I don't cut any of the organs underneath. I'm going to do it on both sides, and then I'm going to cut up the sides. The deeper I go down, the easier it is for me to see everything inside. The more I'm pulling on this skin flap, the more tension I'm given and the easier it is for me to then cut. Now, what I want you to see is, can you see how it is completely sealed off? There is a complete sealant wall right here. That is the backside of the diaphragm. Now, I need to cut through this bone. So I'm going to cut through the ribs, and I'm going to be very careful that I don't just packet it too deep. And now I'm going to cut down the sides of the diaphragm. Right. I'm going to cut up here. What we're going to say. All right, fine. I'm going to cut through the ribs. Yes, it's a little crunchy. And again, the reason why it's crunchy is because this is a fully formed adult. All right, and now I'm going to do the same thing on the other side. I'm going to cut the flaps off, and you can see how cutting through the thoracic cavity allows me to get into, see the lungs and the heart and all that good stuff. Okay, I'm just going to open this bit up. You don't have to. All right, are there any questions for me about how to dissect this? No? Okay. You can, and so now you've got this, and you can see this whole wrap is not a well wrapped. You can see that I'm now in, and I've opened it up and I can see it. Okay, any questions? All right, you may begin. Cut down the middle. There's no talking. Hey, I cannot hear if somebody has a problem if you all talk. So you cannot talk. Just get on and do it. Go. The talking rule does not exclude anyone. If you are talking, I take 10 points off. Okay? I'm not kidding with you. This is just not okay. Okay, how are we doing? So don't be afraid to cut deeply till you get to the bones in the thoracic cavity and then lift up when you get to the abdominal pelvic. Sorry? You can't lift at the thoracic, you can only lift at the abdominal pelvic. Well done. So all the way down the middle, then go to the thighs sideways. So once you've gone all the way down the middle, go to the thighs sideways, then cut the flaps off. Okay? And cut off the sides. Now you can separate this between, you're going too deep, you're hacking, you need to lift up, okay? You can separate it and you can do a cut, then your friend can do a cut. Yes.
Why is it green?
Well, yours have been injected with dye to show the different structures. This is, at one point, there was a rat shortage and these were the only ones I could get. Where are you going? Go that way and then cut the flaps off. Well done, ladies. Okay, lift where you are trying to cut. Give it some, that's it, well done. That's it, well done. Yeah. I find, make sure your fingers don't go past the grippy mark. Lift and give it tension to help it to cut. Again, be even about who's doing what. Make sure everybody gets a turn. Okay.
After we cut open the front, how do you get?
Then you cut just through the rib, through the ribs. No, that's fine now, go horizontally. Okay. The more tentative you are about your incision and your cut, the harder it is. If you are more forceful and determined with your cut, they are, and more like, this is where I'm cutting and you draw it, rather than you're tentative, you will have much greater success. Be definite, be determined. The more you give you a good swipe of the scalpel, it makes a much bigger, better situation. Can you help you? Okay. All right, make sure you're sharing. Yep. No. Put the scalpel in the tray.
How much further do we go up?
Well, you've got to go through the ribs. So, but if you see the diaphragm, stop there, and then I find it easier to cut through the middle first.
Oh, no, oh, yeah.
You have the best one. All right, so there are two different brands of rat, or two different typings of rat. The ones that are yellowy looking and have got fun colors in it have been latex injected to be visual. So you can see where things are. So we're gonna go over. You want to do it? Right. So these ones are actually tougher. And the ones that are not latex injected, these ones are going to they're just a different process of preserving.
Why does ours so like flat?
Because of the back. Back. I don't particularly like these yellowy ones. Oh, that's still better. No, not you, for razor. okay? Is anybody having troubles or, in danger? Because I can hear talking. Yes. So if you got a yellow one, you got it whereby they injected the latex and then they sewed it together so that the latex would then harden and solidify in there. So, you can then see organ structures.
Yeah, we'll check that out.
White pillows. Might still be on.
Oh.
All right, everybody good?
Oh, yeah.
That'll be fine. Good, 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 good. Good. Visually, I'm seeing good. All right, so remember, scalpels must be kept flat in a tray. Do not leave them hanging on the sides. If I see that, I will take points off, both in this lab and the exam. So here we go. So let's start. Gosh, let's start in the thoracic cavity. Mine is not a very good specimen because I can see it's got tumors. This poor rat has lung cancer. Maybe it was a smoker. Not a good idea. Remember, smoking is going to change your brain chemistry, your neurotransmitters, and that the nicotine that's inside cigarettes, inside the tobacco product, is going to change your dopamine receptors. Remember, dopamine is that neurotransmitter that we like because it gives us pleasure and joy and says, great job, do it again. But unfortunately, addiction comes along and it says, hey, what? This is amazing. Oh, wait. And now you've got a narrowing of pleasure, which is what addiction is, because the level of dopamine, the responders are so high and you're not getting the amount of dopamine back again. And so you're always feeling like you're without, and you have to keep doing that addiction in order just to feel normal. Okay. So this rat, not so good. On the sides, I've got some really sad little lungs. These lungs are lobed and they're soft and squishy. You can get your fingers in and give them a feel. So you can feel the difference between the lungs, which are lobed, soft and squishy, and then the structure in the middle, which is like a hard bean-like structure, which is the heart. Remember that the point of the heart is pointing down to my feet, which is the apex, and it's all about the base, not the apex, because the base is where all the tubies connect, okay? So if I were to want to remove the heart from this organism, I could quite literally just take my fingers in here, grab hold of it and give it a little pull. And if I were to do that, I would see that on the outside of the heart, there was a membrane, one of my favorite words, the pericardium, that goes around the outside of the heart as protection. There is also these flappy bits. These are the auricles. Remember, the auricles are the top chambers, the A over V, which is the atria over the ventricles. The auricles are the start of where the blood goes in on the left and the right side. The left side is for the oxygenated blood that's come from the lungs and it's going to get squeezed to the entirety of the body. The right side of the heart is going to be the blood that is coming from the rest of the body that's being used up because cellular respiration, and is trying to now go back to the lungs to have that exchange through the process of osmosis at the alveoli. Okay, happy with me so far? Remember that we've got it. Oh, if I were to cut the heart in half, I would go auricle to auricle, and I would look inside and I would be able to see the septum that divides the left from the right side. And if I was very clever and I looked on the outside of this heart, I would see the coronary arteries that are going to feed this cardiac muscle. And if I'm really clever, I would also see the largest right here, the largest blood vessel that is in the body, which is the aorta, which takes the blood to the rest of the body. Now, this is the flappy bit, which we just looked at previously. This is the diaphragm. And the diaphragm is really important because this is what moves. It pulls down and the ribs go out, which allows for an increase in volume, which decreases the pressure and the air rushes in, because this is Boyle's law, P1V1 equals P2V2. Now, we're going to go down here to the liver. This is the largest gland and structure in the abdominal pelvic cavity that is one structure. This poor rat has a tumor. That is not a gallbladder. They do not have gallbladders, but if they did, I would quite literally lift up the liver and it would be a teeny tiny little sac right here. Now, the liver's job is plentiful. In fact, it's got over 500 functions, but I only want you to know 3. It is for storage. It is for detoxification. It cleans out the blood. And the third thing is it produces bile. Bile is a enzyme that is going to break down the fat. Now, the gallbladder is in us, where it is going to store some of that bile and make it even more concentrated. And if you eat like a double bacon cheeseburger, it's going to eject out so it can break down the really fatty food that you have. There are 3 accessory organs within a digestive system. We have the liver, the gallbladder, and then we have the pancreas. Now, the pancreas is a weird structure in this rat. It is this granular looking structure right about here. Okay, it is a granular structure looking like this. In us humans, it is retroperitoneal. It is underneath another organ. It is underneath the stomach, and its job is to produce insulin to regulate your blood sugar levels. We talked about diabetes, and so now I'm going to move on. All right, lift up your liver, and I want you to see if you can find this J-shaped pouch. It could be hard, it could be squishy, I don't know. Have a lift up of your liver, and on anatomical left side, you should see a lighter pink squishy pouch. It's J-shaped. Yeah, that's it right there. Yeah, that's it right there. Yeah, squishy right there. Yep, that's the stomach. No, this bit right there. Well, you will, this flappy bit with the pancreas. That's the bit right there. Okay. Oh, this is pretty complex. All right, so we'll do a little bit more here on this.
Sorry, where is the part?
The heart is that bit right there. Okay. Yep. So it should be a nice soft. Yours must be on the heart right there. This is your liver. You can see how vascular it is like that. On the left-hand side, to the left of that stomach, there is a hard, flat, there is a hard, long disc. I'm really sorry, these ones that are coloured are really not a pleasant one to deal with. There's that, yes, please. The heart structure is a long yellow, not yellow, it's a Gosh, I hate. These are all you right? Yeah. That's and your screen is just. All right, so there is a flat, long disc. Boys and girls, I'm not kidding you about those 10 points. I'm getting annoyed. Thank you. I did give you fair warning that I was frazzled this morning. This bit right here is our spleen. Remember that our spleen is going to be involved more with the lymphatic system, with immunity. So, so far we've seen the respiratory, we've seen the nervous, we've seen muscular, we've seen respiratory. We're going to look at reproductive in a second. Skeletal, we've seen bone cutting through the jaw, the dentition. L, lymphatic, that's going to be this, it's going to be your immunity, right? Li, integumentary is this, cardiovascular, we saw the heart. Endocrine, when we look at the ovaries, then that will be part of the endocrine because they produce hormones. The rest are going to be in the brain. Notice I'm not getting into the brain. It's just too difficult. And then digestive system is what we're in now. So this is the stomach, and you can see how the stomach is going to continue along and go into the stomach to the small intestine. The small intestine is the longest part of the intestinal tract, and it is this stuff right here. This is all small intestine. And it is small based upon the diameter rather than the length. It is inside this, it contains those villi, those finger-like structures that are going to suck up all of those nutrients. Inside the stomach, there are the rugae, which are the wrinkles, which will expand and allow for grinding. Again, in the stomach, there's chemical and mechanical digestion going on. The duodenum, which is the first portion of the small intestine, is the site for the greatest amount of chemical breakdown because all of the liver, the pancreas, and the gallbladder's digestive juices get dumped into the duodenum. Then we have the jejunum and the ileum, which is the small intestine parts, and then it joins to the cecum. Good gosh, this poor rat has a massive tumor. That's not normal. Now, the large intestine is made up of the, the cecum with the little appendix on it. Remember, the appendix is that vestigial structure that's not really vestigial. It actually does quite a lot for holding our gut flora. There's that bacteria that is really important within our intestines. And remember, we also have vaginal flora. These bacteria are going to help with the absorption of nutrients and the breakdown of certain foods, particularly cellulose. And then it goes up on us to the ascending, the transverse, the descending. It gives a wiggle of the sigmoid, goes to the rectum and then the anus. So this is the large intestine, and the large intestine is large based upon the fact that the girth of it is much wider. The purpose of the large intestine is going to be to absorb water and to form poo. So this is where all the waste is going, and then it comes out and down. All right. Time is not my friend this morning. All right, so what we're going to do is we're going to shift down to the very posterior part of this ventral surface, and we're going to have a look at, there should be some little beads. These are my babies that are in a uterine horn. Now, these babies, boom, 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 this is different for a rat than us because they have multiple uteri. They have multiple uteruses, right? We've only got one. We tend to only have one baby. They tend to have a litter of cups, which is why on their ventral surface, when we saw the teats, we saw that there were multiple pairs of teats. That is because the rat will have many, many babies. So these are the babies, and if I go up the uterus, I'm going to find some fluffy duffy stuff. I'm just going to give it a pause so you can see it. This fluffy duffy stuff is attachment and fat that holds it in place. And then in between, you'll see a little round ball. It looks almost granular, like it's got little dots to it. That is the ovary. Now, this ovary is incredible because this ovary, it's between my fingers here, the ovary is going to contain all the eggs that the rat will ever have, or we as humans will ever have, and it produces hormones, hence it's part of the endocrine system. Now, what we're going to do is we're going to find our ovaries, have a look at it, and then take out a baby. So go ahead, find your ovaries. You can do it on both sides. All right, they're hard. The babies are hard. Don't think that it's a tumor. On either side. You might want to do these even babies. That's the takeoff. Yeah. You you've only just pregnant. Go to the very end of these. And then. Here's yours. I'm gonna say something to your babies. No. Yeah. These babies, yeah, that's the things we've got there. Okay. So, what you're going to do is you're going to take a baby, just rip it with your hands. You don't need to use the scalpel. I'm going to tell you to keep your mouth shut because I'm done talking. And secondly, you don't want it to squirt on you if you pop it by accident and then get amniotic fluid. So make sure you keep your mouth shut. It did happen one time that somebody thought that he was going to be really funny and squirted at his friend, but it quite literally backfired and went, and it squirted in his mouth. Karma, it was great. Keep your mouth shut. So when you take it out, you're taking off the uterus. I want you to quite literally peel it, okay? So as you peel it, you'll be fine. Just wash your face afterwards if you're you're going to peel it like this, and you'll notice that this was the uterus, and it could still be in a little sac. This was a little hard to see because it's so small. So I borrowed a friend's. And I'm going to peel this uterus. Oh, I'm getting fogged up here. I'm peeling this uterus off. And what I'm going to see is that there is a disc, a big flat disc that is right here. This disc is the placenta. And this is connected to the baby via the umbilical cord. And here is the baby. So this is the fetus. This is the umbilical cord, and this is the placenta. So the placenta is attached to the mother and to the baby via the umbilical cord and uterus, right? And this is where the good, the bad, and the ugly are going to get exchanged between the mother and the baby. So if the mother drinks alcohol, that's the ugly is going to get into the baby and cause birth defects. If the mother smokes, that the ugly is going to go into the baby and cause addictions and all sorts of problems. If the mother puts in nice vegetables and nutritious food, that goes in to feed the baby. The waste product that the baby has is going to go back into the placenta, and then the mother's going to extract it and take it away. You should be able to see your own baby and the placenta. Now, what I want you to also notice is that this was their rat baby. This is the actual baby that was in my wrap, and you can see how it is significantly smaller. That is because the gestational age, you can see how there's my baby, and that's my placenta. In fact, the placenta is larger than the baby. And you can see, I'm going to put one on top of the other. You can see the size difference, right? This is my baby, that's their baby. Can you see? Hello. Their baby, my baby. Why are they different sizes? Oh, did you get squirted? Oh dear. They're different sizes because the gestational age is different. When a baby is in the mother's uterus, it will have to take a certain amount of time to gestate. That means to grow to full term. For us as humans, we are 40 weeks or 9 months. If the baby is born prematurely, then it is less than 40 weeks. The rat, however, is much quicker than that. I think it's like 3 weeks. So I don't know, I'm not an expert on fecal rats or how old they are, but as you can see, they're both classified as fetuses. They're both fetal. I cannot determine the age of these or the size of these. They're just what I'm given, right? Does that make sense where I'm going with your clue right here, right now? So the sizing of them is dependent upon how how much time they've been in utero, in the uterus, okay? But these are the same. If you feel it, you can feel that they're actually really quite flexible and wobbly. Why? Because none of their bones have been solidified or hardened, okay? All right, so that was the baby and the placenta that I wanted you to see. The last thing that I want to see is the urinary system. So what I'm going to do is I'm going to shove everything to one side. And what I'm going to see inside, is that there is a kidney bean structure to the left and right of the spine. So here is my kidney bean structure. I'm going to shove everything to one side because this is a definite retroperitoneal organ behind other organs within the abdominal pelvic cavity. It is a bean-like structure. You'll notice it is within a renal capsule. It is covered by a protective layer. You can go in and grab hold of it and give it a little pull. And if you're very clever, you will see that it is attached to a number of tubes. These tubes are going to be, this one that is going down, that is going to be the ureter, which goes to the bladder. And then the other ones that are going into and out of it are the blood vessels. They are the renal arteries and veins. If I were wanting to cut my kidney, which I obviously want to do, I'm going to remember Pearman's silly saying, which is keep the hilus to the sky and cut lengthwise. The hilus being the indented region. I'm going to hold it in my fingers and I'm going to cut it in half and I'm going to look inside. Now, when I look inside, I can see that there is two different coloring. I've got the outer portion, which is the renal cortex. I've got the inner portion, which is the renal medulla. What I remember is that this is just the warehouse. Inside this kidney are the functional units, which are the nephrons. They're the ones that do the work of filtering. And it is specifically in the loop of Henle, which is in the medulla region, that is going to be the purpose for removing and extracting water through the process of osmosis. Now, osmosis is different than diffusion. Diffusion happens in the alveoli, where we have the movement of any molecules, particularly oxygen and carbon dioxide, going from a region of high concentration to a region of low concentration. But with osmosis, there's two key differences. It's water, and it is through a semi-permeable membrane. That is the loop of Henle in this nephron. And that is what's going to take out the water on the one side and pump out the sodiums and all of the other ions, the salts on the other side, making the medulla, that region, the middle region, hypertonic to allow for this osmosis to occur so we can conserve water. All right, let's do a quick review here. You can keep looking at your babies if you would like. Let's just go back through and make sure that we've got everything covered. Run, Mr. Slice. Respiratory. We looked at the trachea, the rings of cartilage. If I was wanting to find the trachea, I would go into the throat and I would look for the one closest to the ventral surface. That is going to be the one that also has those distinct rings of cartilage. Right behind it is the esophagus. I would pick it up and run my thumb underneath, and I could push out the esophagus from sitting underneath. It's a flat, flexible tube. Urinary. We looked at the kidneys. We dissected the kidneys. We talked about the nephrons. Nervous. We talked about the brain. There's 4 lobes in our brain made up of 2 different hemispheres. We talked about how it was, what it did, and the function of it. We talked about the cerebellum, the cerebrum, the brain stem. We talked about all of that in great detail. We talked about the meninges and how they're there for protection. Our muscular. We talked about the fact that there are 3 different types of muscles, right? That's about all we really talked about. R, reproductive. We talked in great detail in the last couple of days about babies. We talked about the male and the female anatomy. We can't talk about the male anatomy because I don't have it here. However, on my rat video, I do actually do a comparison between the male and the female rat. And what you can see very clearly is that when the female rat is pregnant, all of her intestines, which are all nicely wiggled out and in the space of the abdominal pelvic cavity for the male rat, are squished up in order to make room for all of the babies. Very interesting. And then I talk about the scrotum, the penis, and so on and so forth, and semen, and blah, 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 in what we just talked about and in the rat video. All right. S, skeletal. Okay, there's bones and mandible, maxillae, dentition. SL, lymphatic. We talked about the spleen. We said it's to do with immunity. I, integumentary system. We talked about hair and nails made up of keratin. We talked about how that it was protection and the different things that hair does. LIC, cardiovascular. I went through in great detail about the structure of a heart and the blood flow of the heart. I'm going to give you a hint. You need to know, I've actually said this before, you need to know the blood flow, okay? You need to know where things are going in the heart. C, E, endocrine. This produces hormones. Hormones regulate things going on in your body. And we talked about how the pituitary gland was the master gland. We talked about how it was with the ovaries and testes producing hormones. And that hormones are going to regulate the female cycle. C, E, D, the digestive system, my favorite system. We talked about it a lot. I went over it again just now. We talked about it from the start to the end of poop, right? We talked about poop colors and structure, and you did all sorts of things with that. Okay, so Run, Mr. Slice is a great way to understand. Remember the 4 pillars. We talked about it being anatomy dictates physiology. We talked about the Run, Mr. Slice. We talked about the hierarchy of life, and we talked about cellular respiration. Those were our main 4 key pillars that helped generate our understanding of this course. Sorry, I keep looking at my watch. I don't want to go over. And then, oh gosh, there was one more thing. The last thing that we talked about as we went through every single system, we talked a bit about health. So this course is designed as anatomy, structure, physiology, function, and health. Something that could go wrong, but that is not going to go wrong because we can fix it, or we can make preventative actions now so that we don't have things going wrong. Does anybody have any questions? Did you all find your kidneys, by the way? Yes? If you didn't find it now, you can take it out and give it a cut if you would like. When you are finished, I am going to need your help here. I would like you to please bring me your rat, a tray, and I'll clear it up. Put all of your other trash in the trash can. And then we'll go from there. Do you have any questions? If anyone has any questions, come and see me. I think I did a pretty good job of this, but if you've got any questions, let me know. Pencils, you can put them on my trolley, please. Don't put them back in the pot. Trash away.